So, hello guys and welcome back to Excelsior Academy. So, we started with our topic of cell cycle, right? So, in the previous lecture, we talked about different phases of the cell cycle, right? So, what are those? We spoke about G1 phase, S, G2, and M. Okay, so now we know at G1 phase, this is the stage at which all the proteins which is required for the synthesis phase, that is DNA replication, that will be formed in this. In the S phase, DNA synthesis will take place. In the G2, it is making ensure that all the DNA which is being replicated is correct and there is no damage to the DNA, and then only it is going to enter into the mitosis. Okay, now the regulation of this cell cycle. With our current understanding of cell cycle regulation, it has emerged from a convergence of results obtained through experiments on organisms like yeast, sea urchins, frogs, and mammals. These studies have revealed that cell cycle of all eukaryotes is controlled by a conserved set of protein kinases, which are responsible for triggering the major cell cycle transition. So guys, as I mentioned before also, this cell cycle regulation, all the proteins are mainly kinases. Okay, now why kinases? Because by phosphorylating, either they are going to inactivate the molecule or it is going to activate. Okay. So, for example, there are three distinct experimental approaches which was carried out to identify the key molecules responsible for the cell cycle regulation. Now, let's take the first example. The first example was taken from the frog oocytes. Now, guys, this is important. Okay, ये exam में आ चुका है. Donor oocyte. So here, what we see is that you have got progesterone add किया हुआ है. So जिसकी वजह से क्या होगा? Directly, okay, it is jumping from G2 to the M phase. Now, what is happening in the M phase? As you know, right? So meta phase rahega and the chromosomes will be aligned. Okay. Now, next what they are doing is, so these oocytes are arrested in G2 phase of the cell cycle until hormonal stimulation triggers their entry into the M phase of the meiosis. Okay, is this much is clear? Like when we say when we say that the cells are arrested in G2 phase, what does it mean? Yes, Krishita, what is the meaning of that? G2 phase arrested?
That is it is yet to undergo the mitosis phase.
Yeah, so it means that if the stimulation is not given, then the cell will be forever in the G2 phase. Okay, so what they said is that donor oocyte is there. So first it was arrested in the G2 phase, and with the help of the progesterone, it was stimulated to enter into the mitosis phase. Okay? Now, so oocytes arrested in G2 phase could be induced to enter M phase by micro-injection of cytoplasm from the oocytes that had been hormonally stimulated. So what they're saying over here is that, let's say that this is the donor oocyte which is arrested in the G2 phase. Okay. Now they have taken, they have given the progesterone as a stimulation so that from G2 to M phase it passes. Now, if I take the cytoplasm of this cell, okay, I'm saying that I have taken the cytoplasm. Okay. Now, what they have done is with the micro injection, they have taken the cytoplasm and injected into the another cell. So what will happen? Even that cell is now stimulated to go into the M phase. Did you get it? So here there was no such additional stimulation was given, but the cytoplasmic factor which was taken from here, it was transferred to the recipient, and that induced this particular oocyte to undergo the mitosis. Did you get it over here? What is the experiment? So experiment is they have taken the cytoplasm, the donor oocyte is there, arrested at the G2 phase. Okay. This is also arrested at G2 phase. But in this case, in the case of the first, they have added progesterone in order to stimulate so that the cell undergoes G2 to M phase transition. And now they have taken the cytoplasm over here, that is introduced into the second oocyte. Now the second oocyte, which was again arrested at G2 phase, got stimulated and entered the M phase without any additional stimulation. उन्होंने क्या किया है, सिर्फ cytoplasm यहाँ से यहाँ लिया है. Okay, did you get the experiment here? Clear? Tanvi, is it clear?
Yes.
Okay, cool. Okay, so it thus appeared that cytoplasmic factor which is present in hormone treated oocyte was sufficient to trigger the transition from the G2 to M in oocytes that had not been exposed to the hormone earlier. Now, because the entry of oocytes into meiosis is frequently referred to as oocyte maturation, that cytoplasmic factor was known as MPF, maturation promoting factor. Okay, so whatever it is, now we know that it is known as maturation promoting factor. Okay. Now, further studies showed that the activity of MPF is not restricted to the entry of oocytes into meiosis. It is also present in somatic cells where it induces entry into M phase of the mitotic cycle. Rather than being specific to oocytes, it is thus appear to act as a general regulator of the transition from G2 to M. So now what we have learned, so maturation promoting factor is one of the key responsible protein which is going to give the stimulation to transition from G2 to M. Okay, so this is the first regulator to remember. Now let's talk about the second experiment. So what was that first experiment? It was about the frog oocyte. Now in the second experiment, the genetic analysis of the budding yeast. So they have identified a temperature sensitive mutants which were defective in the cell cycle progression. Okay, now these temperature sensitive mutants, they are also called CDC for cell division cycle mutants, was that they underwent growth arrest at the specific points in the cell cycle. When you say temperature sensitive mutant, it means that if you increase the temperature of the surrounding, the yeast might die or it will replicate. Okay, so depending on the conditions, they have made the mutations. Now, for example, a particularly important mutant designated as CDC28, that is cell division cycle 28, it caused the cell cycle to arrest at the start, indicating that CDC28 is required for passage through this critical regulatory point in G1. Did you get the point over here? So we have a CDC28 mutant, which is causing the cell, the budding yeast, to arrest at the start. So what was start, guys? Do you remember in the previous lecture we have discussed? So in the cell cycle phases, when we spoke about it, mitosis, G1, S and G2, right? This is around 11 hours. This is 8, 4, something like that, right? 12 plus 11 is 23 and 1. Yeah. So in the G1 phase, here we have this regulatory checkpoint which is known as start in the case of the budding yeast, or it is also known as the restriction point. Okay, so now we are saying that there is a mutant CDC28 which is arresting this particular cell cycle at this point. Okay, start. So which is saying that CDC28 protein is important for the passage through G1 so that it can enter S. Now, how this experiment is being conducted? So let's see over here, we have got, so normally the mother cell is there, it enters the cell cycle, it goes to G1 to S to G2 to M, and finally you get a daughter cell. Okay. Now, when the temperature is not permissible, it means it is not in favor of the cell, what will happen? This checkpoint is not allowing them to go into the S phase. Able to follow, Akshaya?
Yes.
So when I say temperature sensitive, what does it mean? It means the temperature has resulted in the denaturation of your CDC28, because of which now there is no checkpoint and the cell cannot enter to the S phase. Okay. So the mutants of CDC28 or to S-POMB cell cycle both in G1 and at the G2 to M transition. Comparative analysis showed that S. cerevisiae CDC28 and S. pombi CDC2 are functionally homologous genes required for passage through the start as well as for the entry into mitosis in both species of the yeast. And these are nothing but they encode protein kinases. Okay. Now, in case of eukaryotes, it is known as CDK1. What is CDK1? It is cyclin dependent kinase 1. Okay. So, which is the second protein now we have discussed? It is the CDK1. अभी guys कैसा होता है ना, यहाँ पे थोड़ा सा confusion शुरू होगा. So first what we have discussed is MPF. जिसका काम था transition from G2 to M. Okay. The second protein is CDK1 in eukaryotes, CDC28 in case of Saccharomyces cerevisiae, and this is CDC2 in the case of Saccharomyces pombi. Okay, they all are same. Now they are responsible for the transition from G1 to S phase and also G2 to M phase. Okay, so till now what we have discussed, this is a summary. Okay, so far so good, guys. Able to follow, Shruti? Okay. Because here so many proteins we are gonna learn, and it's all like CDC, CDK1 and this and also that's why the confusion will come. Now the third experiment, it was carried out in the sea urchin embryos. So following the fertilization, these embryos go through a series of rapid cell division. So as we know, the moment the embryo is formed after the fertilization of egg and sperm, what happens? Rapidly, so many times the division is taking place without the growth. Okay, so what happens? The content of the cytoplasm is getting reduced. Okay. If the cell is there, it is divided into two. It means totally half of it has gone. It's not that it is similar to your parental cell. Now, studies with protein synthesis inhibitors had revealed that entry into M phase of these embryonic cell cycles requires new protein synthesis. So these proteins accumulate throughout interphase and are then rapidly degraded towards the end of each mitosis. So they are the proteins known as cyclins. Okay. So as two proteins are there, so they are named as cyclin A and cyclin B, and they suggested that they might function to induce mitosis. Okay. So it means what? Let's see over here in this figure. So as we see here, since we said it is for the mitosis, so we are going to check the level of the cyclin. Okay, so what do we see here? As interphase is increasing, okay, you can see that the concentration of cyclin is also increasing. And in the mitosis, it is the maximum. Towards the end of the mitosis, it starts declining, right? And again, when the cell enters the interphase, again it is increasing. During the mitosis, it's the maximum, okay? And again, it is rapidly degrading towards the end of the mitosis and the cycle continues. Clear? So those are known as the cyclin A and cyclin B. Guys, stop me, okay, and ask if any point you're not able to follow, because so many proteins are there and everything we need to remember. Now again, coming back to the MPF. So in the case of the MPF, when we said that it is your maturation promoting factor, which is the transition, right, from G2 to M. That was the first experiment which we learned from the frog oocyte, right? So maturation from G2 to M, it is transition from G2 to M. So now we know that it is the key regulator of the cell cycle and it has got the two subunits. Okay. Now what are those two subunits? MPF is made up of cyclin B and it is made up of CDK1. So where cyclin B act as a regulatory subunit. Clear? and CDK1 is your catalytic subunit. So MPF getting activated means the concentration of cyclin B is also increasing. Now, when cyclin B interacts with CDK1, the complete MPF is formed and the transition is taking place to G2 to mitosis. Clear? Krishita, is it clear till here? So far, what we have learned? Okay.
Yes.
Cool. Now, the regulation. This is important. This has come for the exam. Okay. So the regulation of MPF is by phosphorylation and dephosphorylation of CDK1. Throughout cell cycle, it is the phosphorylation only, okay, which is going to make it active or inactive. So exam में अगर कुछ question आता है related to the cell cycle and saying that what kind of modification is taking place or whatever it is always a phosphorylation with respect to cell cycle I'm talking, okay. Okay, so in the case of the mammals, cyclin B is synthesized and it forms a complex with the CDK1. Just now we saw during the G2 phase. Now, as these complexes are forming, CDK1 is phosphorylated at two critical regulatory positions. Now, what are those? So, that is occurring at the threonine 161, okay, which is required for your CDK1 activity. And the second one is tyrosine 15 and threonine 14 in the case of the vertebrates. So there are two phosphorylation taking place, threonine 161 and tyrosine 15. Now, phosphorylation of tyrosine 15 is by another protein kinase known as V1, which inhibits the CDK1 activity and leads to the accumulation of inactive CDK1 cyclin B complex throughout the G2. Okay, I just hope that you guys don't get confused. So just now we talked about cyclin B, right? And we mentioned about the CDK1 activity. Okay, so if phosphorylated at 161, threonine 161, it is active. Okay. Now, if phosphorylated at tyrosine 15, which is done by V1, it is another protein kinase, this is inactive. Clear? Okay. And we have got here another, sorry, this I didn't mention this one. This is also present at 14. Now, this is the inactive structure. So, in order to bring about the activation of this CDK1 and cyclin B complex, the dephosphorylation is required of your 14 and 15. ये कौन करेगा? Phosphatase known as CDC25. Okay, I hope I have the figure so that it becomes more clear. Yeah, here. This is, guys, really very important. Okay, I'm telling you again and again. This has come for the exam. Okay, so we have got cyclin B. Cyclin B goes and binds with the CDK1. Now there are three sites for your phosphorylation. So V1 is going to phosphorylate, and what we get? Threonine 161, tyrosine 15, threonine 14. Okay, now this is inactive. This stage is still inactive unless until CDC25 comes and dephosphorylation is happening of your threonine 14 and tyrosine 15. Then only this is active. This is your active MPF, which will bring about the transition from G2 to M. Clear? So it means now the cell is entering into the mitosis. Now, once this is done, towards the end of the mitosis, the cyclin B will start degrading and CDK1 will get phosphorylation and whatever, and it is going back to the cycle. Clear? Is this cycle is clear to everyone? Akshay, Tanvi, is it clear? Should I repeat? Do you want me to repeat the cycle again?
Please repeat.
Okay.
Ma'am?
Huh. Yes, yes, Krishita, you were saying something?
Yes, ma'am. Please repeat, ma'am.
Okay, okay, yeah. Okay, so don't look over here. Let me show it here. Okay, so you have got MPF, which is made up of your cyclin B, and you have got CDK1. Okay, so this is your MPF, maturation promoting factor, but this is nothing. It is not active, right? So to make it active, there is series of steps. So first step is that V1 as a protein kinase will come and it is going to phosphorylate your CDK1 at threonine 161, tyrosine 15, and threonine 14. Okay. Now, still this is inactive. In order to make it active, we have got a dephosphorylation step or phosphatase known as CDC25. Okay. So CDC25 क्या करेगा? It is going to remove your threonine 14 and tyrosine 15. So these phosphates are being removed. Now, the cyclin B and your CDK1 along with your threonine 161 phosphate, it is your active MPF. Now this will enter the mitosis. Okay. Now towards the end of the mitosis, what is happening? As you see in the previous figure, we showed that towards the end of the mitosis, the cyclins levels are decreasing. Okay. So what does it mean? Then the cyclin B degradation is happening. Now, if you remember in the case of mitosis, we have something called APC. What is APC? Anaphase promoting complex, right? So this is going to ensure that the cyclin level is now degrading. Cyclin B is degrading. Now, cyclin B is not there. So what will happen then? Your CDK1 will become inactive. And when again the mitosis step is coming for the new cell cycle, the level of cyclin B will start again during the entry phase and it will combine with the CDK1 will become an active MPF so that it can enter the mitosis. Clear? Any step which is not clear, just go through once by yourself and see at which stage you're not able to understand. Krishita, would you like to explain? Because once you yourself explain, then it becomes clear whether you have understood or not. Just try explaining to others, and let's see how much you have understood. Okay, so I'll be there. I'll explain in between if required. Yeah.
Ma'am, I'll have to go through it once.
Yeah. Take your time and just start then.
So there is this CDC 25, which, I mean, the CDK1 and cyclin B, these are present right at ma'am, I'll have to go through it once. I won't be able to.
Yeah, start from here.
No, ma'am, not very confident about it.
Okay, anyone else who would like to try? Akshaya, Shruti or Tanvi? There's nothing wrong. You can just try. Shruti, would you like to try?
Okay. 1 second. Okay. So cyclin B and CDK1 are CDK1 is the catalytic component, and cyclin is the one that activates it. So at this particular point at the top.
No, Shruti. Not
Yeah.
like CDK1 is different subunit, cyclin B is different. Okay, cyclin B is your regulatory subunit. CDK1 is
Yeah,
your
yeah.
catalytic subunit. Okay.
They're both different. They can only work together.
Yes.
Yeah. Yeah. So actually, they start out as dephosphorylated, and after phosphorylation, which is done through V1, cyclin B, cyclin B remains connected, and CDK1 gets phosphorylated at three positions: threonine 14, tyrosine 15, and threonine 160. So and then there's a dephosphorylation step, which is mediated through CDC25, and that causes the dephosphorylation of threonine 14 and tyrosine 15, which is the complex that is fit for transferring, for causing mitosis. And after that, after mitosis, cyclin B degradation gets started, and therefore CDK1 is again inactive, but then it's still phosphorylated at threonine 161, and that phosphorylation is also removed, and finally it is back to the start where cyclin B is not connected, but then present in the cell due to other factors. And when it connects, that's when the cycle can start all over again.
Okay, so when it connects with the cyclin B, it means the cell has completed its interface. See, if you see the figure, aise tha na, something like this. So in the interface, it is keep on increasing. Okay? And when in the mitosis, it is maximum. And then it starts falling back. Right? So falling back means this is what? Cyclin B degradation. Okay?
I forgot about anaphase promoting complex.
It's okay. So APC is responsible for the degradation. Okay, cool, good. So the same thing over here, what just now we have discussed. So CDK1 activity triggers the degradation of cyclin B, which occurs as a result of ubiquitin mediated proteolysis. And this proteolysis is mediated by ubiquitin ligase called the anaphase promoting complex cyclosome, which is activated as a result of phosphorylation of CDK1 and cyclin B. So did you see CDK1 cyclin B is important to form an active MPF so that the cell can undergo mitosis, but it also activates APC, which in result is going to destroy cyclin B, and it is going to inactive your MPF. So this will help in bringing the cell out of the mitosis and undergo cytokinesis and back to return to interface for both the daughter cells formed. Clear? Now, the families of cyclins and the cyclin dependent kinases. So, CDK1 controls the passage through the start as well as entry into the mitosis in the yeast. Just now we saw, right? So, it does so in association with the distinct cyclin. So, cyclin dependent kinase, from the name itself, it's clear that unless until it is bound to different types of cyclins, it cannot function. Okay. So J2 to M transition is driven by CDK1 in association with your cyclin B, which is also known as, these are the different names. It's okay, don't worry about this, just remember cyclin B. Now passage through start is controlled by CDK1 in association with distant class of cyclins called G1 cyclins. CDK1 then associates with different B-type cyclins which are required for progression through the S phase. Okay, so right now what we are saying here is that G2 to M is your CDK1 plus cyclin B. Clear? Now, for G1, we have CDK1 plus cyclin G1. Clear? So these multiple members of the CDK family then associate with the specific cycles to derive progression through the different stages of the cell cycle. So as name says, cyclins, it is understood that they are responsible, for different phases of the cell cycle. Okay, so this is the summary. Now, for example, progression from G1 to S is regulated by CDK4, 6 and 2 in association with cyclins D and E. This is something, guys, if possible, try to make a table or write it in such a way that you remember this entire slide. Okay, so from G1 to S, it is your CDK4. G1 to S, it is your CDK4, 6, 2 plus cyclin D and E. Okay, here again, cyclin 4 and 6 are with the D-type cyclins. Okay, so they are responsible for passing your restriction point or the start point, which we have mentioned. Now, E-type cyclins are then expressed. CDK2 is with E. 4 and 6 are with cyclin D. Okay, then they are required for the transition and initiation of your S phase. CDK2 with A are then in the S phase, which will function through the G2. Now, the initiation of mitosis and passage through the M phase is again with the CDK1 in complex with A and B cyclins. Okay, should I summarize for you or is it clear? Okay, I'll just summarize for you guys. G1 to S is CDK 4, 6 and 2. Okay, CDK 4 is 6 is with cyclin D. CDK 2 is with your cyclin E. अब इनका काम क्या है? CDK 4, 6 cyclin D is passing the restriction point. CDK 2 and cyclin E is at the later transition, जहाँ पे आपका initiation of DNA synthesis will start. Okay, then now you are in the S phase. G1 to S transition हो गया. Now you are in the S phase. S phase में CDK2 plus cyclin A. ठीक है? Now this will help in the transition from S to G2 phase. G2 phase में, as you remember, CDK1 है and cyclin B है. But towards the starting, we have cyclin A as well. Okay. I know it will take time, guys, but you have to remember this thing. ठीक है? नहीं समझ रहा होगा तो then you have to buy hard this thing. Okay, so this is how you can remember also, if you feel that it is better to remember like this. So go with the mammalian one, CDK4, 6 with the D, then CDK2 with the E, CDK2 with A at the later stages of the S phase, and then it enters the G2, where G2 is with the CDK1 and cyclin A. Then in the mitosis, it is CDK1 with the cyclin B, where it forms the maturation factor, promoting factor. Okay, so this is better that this figure you study. Try to remember. Okay, so in addition to regulation of the CDKs by cyclins and phosphorylation, their activities are regulated by the inhibitory proteins also, which is known as the CKIs. So the CKIs, what are CKIs? They are the cyclin dependent kinase inhibitors. In mammalian cells, 2 families are there which is responsible for regulating the CDKs. For example, the members of the INK4 family, which bind to inhibit 4 and 6. Okay, so they inhibit the progression through the restriction point in G1. So इनका inhibitor कौन हो गया? INK4. In contrast, members of the CYP and KIP family inhibit complex 2. Okay, thereby inhibiting the entry into the S phase and progression to G2 phase of the cell cycle. CYP and KIP inhibitors. Here you can study like this. Once you study this, so G1 क्या है आपका? Restriction point cross करने वाला है cyclin 4, 6 with the cyclin D. So who is the inhibitor over here? INK4. Now, if you have CDK2 and cyclin E, so that it can progress throughout the S phase and enter the G2, then it is your CYP and KIP family. Guys, Is it clear this much? Akshay, able to follow? Tanvi? Same thing. The table is indicating about the inhibitors of the cyclins. Okay. Okay. So, yeah. So now the growth factors, okay, the growth factors which is going to regulate your G1 CDKs. So proliferation of animal cells is regulated largely by the extracellular growth factors, which control the progression of cells through the restriction point in the G1. Now see guys, this is also important to understand. एक cell है आपके पास and here you have got the growth factors, okay, because these are going to determine whether the cell should undergo the cell cycle or not, or it should remain in the G0 space. G0 where there is no stimulation, no growth factor, so there is no information given to the cell that it should undergo the cell cycle or not. Okay, so depending on the growth factors which is present in the outside surrounding, the cell is entering into the different phases. So this control of cell cycle progression by extracellular growth factors implies that intracellular signaling pathways stimulates downstream of the growth factor receptors. Ultimately, what is happening? The regulating the components of the cell cycle machinery. So here the growth factor which comes and binds with the receptor, it is going to activate the downstream cell signaling components, right? And that is making ensure that the cell is undergoing different phases of the cell cycle. For example, let's talk about the D-type cyclins. So cyclin D synthesis is induced in response to the growth factor stimulation in part by signaling through the RAS-RAF-MEK-ERK pathway, and cyclin D continues to be synthesized as long as the growth factors are present. So guys, now I hope you remember the RAS-RAF-MEK-ERK pathway. Okay, so what is happening here? So the growth factors will come and bind to the receptor, right? And what receptor here we are talking about? The tyrosine kinase. So when the growth factor comes, the dimerization of the tyrosine receptor happens, then autophosphorylation happened, then those phosphorylated cytosolic domain of the tyrosine gets recognized by the SH2 domain of the proteins, right? Now that is going to activate your RAS protein then. The RAS gets exchanged. Instead of GDP, you will have now GTP. Now this activated RAS GTP is going to activate your RAF. RAF is going to activate MEK. MEK is going to activate ERK, right? So they all result in the cyclin D synthesis, and this will keep on till the growth factor is present. Okay. Now cyclin D binds to your CDK4 and CDK6. So what is the function of CDK4 and CDK6? To pass through the restriction point or pass through the start point. Okay. Now cyclin D is also rapidly degraded during G1 as a result of ubiquitination of the ABCC ubiquitin ligase. So its intracellular concentration rapidly falls if the growth factors are removed. Okay, so ABC is responsible for declining the concentration of your cyclin D. Thus, as long growth factors are present through the G1, the complex of CDK4, 6 cyclin D will drive through the restriction point. So, यहाँ पे कौन सा आपका cell signaling pathway use हुआ? RAS-RAF-MEK-ERK pathway. Okay. On the other hand, if the growth factors are removed prior to this key regulatory point in the cell cycle, then cyclin D will rapidly fall and it will unable to progress through the G1 to the S phase. यानी कि क्या सेल वापस आपका G0 stage में चला जाएगा, ठीक है? And if see, it's very simple to understand. Your cell is there, you have given the growth factors. So it prepares the cell to enter from G1 to S. But immediately, as we know that G1 to S is around 11 hours, right? So as the cell is progressing and you remove the growth factors, what will happen then? The concentration of the cyclin D will fall off and the cell gets arrested at the G1 only directly. Clear? So the inducibility and the rapid turnover of cyclin D1, it thus integrates growth factor signaling with the cell cycle machinery, allowing the availability of extracellular growth factor to control the progression of cell through the G1. Okay. Now, cell cycle regulation by RB, cyclin D and E2F. We are just increasing the complexity of the cell cycle regulation. Okay, RB is your retinoblastoma. So, mutation to cyclin D contributes to the development of variety of human cancers like lymphomas and the breast cancer. Retinoblastoma, it is one of the first identified as a product of gene responsible for retinoblastoma. So, it is what your eye-related cancer. So, it's a rare inherited childhood eye tumor. So, it's actually a tumor suppressor gene whose inactivation leads to tumor development. So tumor suppressor gene मतलब ऐसे ही active होना चाहिए. So once it becomes inactive, it means tumor will form. Whereas oncogene proteins such as RAS and cyclin D, they derive cell proliferation. The proteins encoded by many tumor suppressor genes act as brakes that slow down the cell cycle progression. Okay, so now you're getting it over here. Let's first understand what is happening. So RB is your tumor suppressor gene, yani ki it should remain active, okay? Because it is going to suppress the growth of the tumor. Now, once it becomes inactive, then the cancer is happening. Whereas RAS and cyclin D are your oncogene proteins, यानी कि उसको normal ही रहना चाहिए, okay, so that they can undergo cell cycle progression. And tumor suppressor genes like RB and the inhibitors like ENC4 CDK, which is for your CDK4 and 6, right, to which cyclin D binds, they're acting as a brake. यानी कि क्या जितना जरूरत है उतना ही आपका cell cycle progression होना चाहिए. More than required leads to cancer formation. Is this slide clear, everyone? Krishita, is it clear? Okay. Now, I'll go slowly, okay? Don't worry. Let's understand this and let's remember from here itself, okay? So the activity of RB protein is regulated by changes in phosphorylation as cells passes through the cycle. In particular, RB becomes phosphorylated by the activity of CDK4/6 cyclin D complex as the cell is passing through the restriction point. So we know CDK4/6 cyclin D is responsible for the transition from G1 to S, यानी कि ये आपका restriction point जो है, start point है, उसको cross करने में help करता है, ठीक है? So this is responsible in phosphorylating your RB. Okay, so this is your RB is now phosphorylated. Clear? Now, in its under phosphorylated form, it binds to E2F family of transcription factor, which regulate expression of several genes involved in cell cycle progression, including the gene encoding cyclin E. Okay, so E2F bind to its target sequences either the presence or absence of the RB. Okay, so if suppose there is no phosphorylation, then RB binds to E2F. E2F is responsible for many gene expression. Now, RB will act as a repressor so that RB-E2F complex suppress transcription of E2F related regulated genes. Okay, we are going very slowly. Okay, this is the complex suppresses the regulated genes. Okay, now, okay, I'll just say once again. See, what is happening is that RB is there. It is in association with your E2F. Okay, it means no gene expression is happening. Clear? Okay, this much is clear, I believe. Okay, now RB is getting phosphorylated. इसको phosphorylate कौन करेगा? CDK4/6 cyclin D complex. Okay, now once it's phosphorylated, RB results in the conformational change and it is going to get dissociated from E2F. तो ये यहाँ से अलग हो गया है. Okay, now E2F is free. It will activate or it will result in the gene expression. So RB act as a molecular switch, which will convert E2F from a repressor to an activator so that the genes required for cell cycle progression are synthesized. Okay. Yes, guys, able to follow? Should I repeat again? I'll explain once again. Okay, can you see this wide screen?
Yes.
Okay. So what I'm saying here is that cell cycle progression. Okay, we are discussing cell cycle progression. So this much is clear to everyone, I believe, that CDK4 and CDK6 complexes with your cyclin D. Okay. This is responsible for the transition from G1 start point to enter into the S phase. Now, during this, more regulations are there, and that is your RB. RB is your retinoblastoma. It's a tumor suppressor gene. यानी कि ये active रहना चाहिए so that cancer ना हो. ठीक है? Now, this is always bound to your E2F, which is responsible for which is acting as a activator for your transcription factor required for your cell cycle progression genes. ठीक है? So अभी क्या है G1 phase में this is inactive. Now, once the cell is in the G1 phase, CDK4/6 cyclin D is active. This will go and phosphorylate your retinoblastoma, which will result in the dissociation of RB from the E2F. Correct. Now E2F is active. It will result in the gene expression of the proteins or the enzymes required for your cell cycle progression. Now it is clear. So retinoblastoma without phosphorylation keeps your E2F inactive. After phosphorylation, this undergoes conformational change and E2F becomes active, and it goes for the gene expression. Clear? Yes, guys, I need some answer, then only I can go ahead, because this point is really very important with respect to your exam. Is it clear? It's okay that you don't able to remember, but I just want that you should be able to understand the concept. Okay, that is more important. Same thing. RB bound to E2F, there is no transcription. CDK4/6 cyclin D complex comes, phosphorylates your RB. RB is taken and it is now E2F is your free, so the transcription is happening. Okay. So this is a regulation between the RB, cyclin D and E2F. Now entry into the S phase. So progression through the restriction point and into the S phase is again mediated by the activation of your CDK2 in complex with cyclin E, okay, which is stimulated by E2F following the phosphorylation of RB. अब जैसे ही phosphorylation of RB is happening, E2F is getting activated, it is also activating your CDK2 cyclin E complex, which will result in passing the restriction point and entering into the S phase. Okay. So in addition, the activity of CDK2 cyclin E is inhibited in G0 or early G1 by the CDK inhibitor P27 growth factor. Clear? CDK2 cyclin E आपका inactive है at the start. That time your CDK4 and 6 is active. Now who is making them inactive? Your CDK inhibitor P27. अब तक हमने कितने inhibitor पढ़ लिए? 3. KIP, CYP and KIP family, INK4 family, and P27. Okay. So this is signaling via the same pathway, RAS, RAF, MEK, ERK pathway, and PI3 kinase, which reduces both the transcription and the translation of P27, lowering the levels of P27 within the cell. Now, once CDK2 becomes active, it will bring about the complete degradation of your P27, जो आपको inhibit कर रहा था आपका, ठीक है? CDK2 cyclin E inhibit कर रहा था. So once CDK2 is getting activated, यानी कि इसका inhibitor का level concentration जो है वो नीचे जाएगा. Now, this positive autoregulation further activates your CDK2 cyclin E, which also phosphorylates and inactivates your APC, which will prevent the cyclin E degradation. And then only it is going to pass from S and enter into the G2 phase. Okay, I'll repeat once again. So till now what we have seen, E2F is free, okay, it is activated. Now it is responsible for the transcription of many genes which is required for the cell cycle progression. Now following this, your CDK2 and cyclin E is getting activated. Okay, so CDK2 cyclin E is now what it is going to do when this is active, it is going to inhibit the production or the the formation of its inhibitor P27. यानी कि क्या CDK2 is increasing, its inhibitor is declining. APC-C complex is also declining, so that cyclin E is present. Okay, and the cell is now transitioning from S to G2 phase. Okay, so now we have seen like how from G1 to S to G2 we can enter. Same thing. So you have got the P27 CDK2 cyclin E. P27 is the inhibitor. So once this inhibitor is removed, okay, so how it is going to do? It is self, yani ki CDK2 cyclin E, it is going to phosphorylate. So 27 is going to removed, right? So P27 आपका inhibitor जो है वो चला जाएगा, resulting in the CDK2 cyclin E का concentration level up हो जाएगा, which will again inhibit the APC complex, so that the cyclin E is not degraded, and hence you have got sufficient amount of CDK2 and cyclin E. Clear? Is the figure is clear to everyone? The transition to S and transition from S to G2 is clear? Akshaya?
Yes, ma'am.
Cool. Okay, yeah, that's all. That's all, guys. So today what we have discussed is, let us just summarize. So we have spoke about the CDKs, types of CDKs and the cyclins, right? How entry G1, how it is entering through the G1, G1 to S, S to G2. Okay, so here it is your CDK4 and 6, here it is your CDK2, 4 and 6 is with your cyclin D, CDK2 is with your cyclin E. Okay, then how RB and E2F are playing important role in regulating the cell cycle. So guys, so far so good, I believe. Do you find any difficulty in understanding, or was I going very fast? Because this is really very important. Exam में आता ही आता है. Okay. So I'll be stopping here now then.